So let's go ahead and pick up where we left off. So the day will move briskly because the exam is going to cover chapters 3 and 4. So we have a little bit of coverage to do on 4, and then at the end of 4, that'll be the cutoff. Okay, so we left off with the energetic organelles, right? Mitochondria being the top of those. And so when you take a look at the structure of mitochondria, basically you have a double membrane system, similar to the nucleus, right? So you've got an outer and an inner membrane. You got space in between those outer and inners, and you got the fluid portion in the inside, which is the matrix, right? So that's basically the construction of it. But the inner membrane is actually a little bit different. So it's actually folded into these sort of flaps that are called cristae. And the reason why you have those folds in there is to increase surface area, right? Because that's where all the enzymes are that are actually making ATP. So the idea is the more you have, the more ATP you get, which is a good thing, right? And so this is kind of the other thing about mitochondria. We're going to take a look at its structure here in just a second, is that they have their own DNA, right? Which is kind of odd, right? Because DNA was our definition for life, right? If you're a living thing, you have DNA. If you have DNA, you're a living thing. It kind of works back and forth. So if mitochondria has its own DNA, then what does that say about mitochondria, right? That it was basically alive, right? So that's kind of what it suggests. So it's kind of like a little bit of a head scratcher. And so we'll kind of circle back around to that again. So hang on to that one. We'll cover that one again. Then we have the structure, right? So here you can see the structure. You can see the ribosomes. Basically, there's little outpost ribosomes that has its own little pot of ribosomes, if you will. And then you can see your outer membrane here, your inner membrane, which is folded up. This is actually a little looser than usual to kind of show you the structure of it. And then, of course, you have your inner membrane space, which is this area here and in between here. So you can kind of see the inner membrane space actually kind of goes into the center portion of it. And then the matrix is actually the part that's in here. So in the center piece of it. And the reason why that's important is because you basically have this division of location, and you have different activities happening. So like when you're getting into the cell respiration process, different parts of the cell respiration process are occurring at different locations of the mitochondria. Like there's different chemical reactions that are happening in the matrix. There is a different set of things that are happening in the inner membrane space. There's different things that are happening in the actual inner folds of the cristae itself. So this is kind of gives you not just sort of an organizational sort of a thing, but it also gives you sort of a division of labor, kind of creates stations of labor within the mitochondria itself. And this is a really good transmission electron micrograph here. You can see it's very distinct, one of the more distinct organelles actually we have in the cell, because there's nothing really that looks like a kidney bean with tiger stripes. So you can see very clearly, right, those little folds of the cristae on the inside, and there's a lot of them. And so those are all busy making ATP, which is a good thing. So that leads us then to chlorophyll. And I don't want to get too much into the mitochondria, right? Because they got their own chapter to break down. That's what chapter 7 is. It's pretty much all of the cell respiration activities that are going on inside the mitochondria. Chloroplast also has its own chapter, chapter 8, right? So I just kind of want to sketch these guys out a little bit and move on, because we got an entire chapter, and that's part of our take-home activity. Okay, so organelles are similar in the sense that they also have two membranes similar to the mitochondria. The only difference is instead of doing cell respiration, they're doing photosynthesis, right? So they're loaded with chlorophyll, which is basically your light absorbing pigment. And that's basically surrounded by two different membranes. Now, the membranes in chloroplasts are also folded up, also to increase surface area, only they're folded up into structures that we refer to as thylakoids. So thylakoids are like little membranous, they're almost like little discs. They're like a little membrane sac that's kind of flattened, kind of like a beret, right? But you have typically several thylakoids. So like an individual thylakoid would be like one of these little stacks. And generally speaking, you have multiple thylakoids sort of stacked up on each other. And the whole stack itself is basically what we refer to as a granum, grana for plural, granum for singular. And then also when you have multiple grana involved, these are interconnected with these little membranous bridges. So the entire system of grana are all interconnected to each other. And so the reason why you have these little thylakoid stacks is because in the membranes of the thylakoids are the enzymes for photosynthesis. So all the photosynthetic enzymes are going to be embedded in the membranes of the thylakoids. Again, the same idea. The more membrane you have to embed your enzymes in there, then the more photosynthesis you're going to be able to do, which means the more energy you're going to be able to make. Okay, so it's all about output and productivity. Again, we see the same theme. Chloroplasts also have their own DNA. The same questions arise. Are you alive? Right? So you have the same basic set of questions. We're going to cover that one here in just a second once we finish up with the chloroplast, and we'll take a look at that. So it's kind of what the chloroplast structure looks like. So here you can see you've got the outer membrane, you've got the inner membrane, and then that folds into these little membranous stacks here. These are the thylakoids stacked on top of each other. And you can see these like little bridges in between, these little membranous bridges that sort of connects one stack to another. So everything is interconnected. And this is basically a transmission electron micrograph of a real chloroplast. So here you can see one grana, there's another one there, there's a little small one right there, there's two small ones right there, but you can see how they're all interconnected by these little threads, these little membranous bridges that are connecting one system of stacks to another system of stacks. So everything is all interconnected. And then the fluid space in between these little stacks of grana is basically called the stroma. So this is the fluid portion. So like all this open area, like right in here, that's all stroma. Okay, so just like the matrix was the fluid region inside the mitochondria, the stroma is the fluid region inside the chloroplast. Now, let's kind of circle back around to this DNA idea, right? So remember when we talked about bacteria, they had two sources of DNA, right? They had their nuclear DNA. Well, they don't have a nucleus, right? But they have their nucleoid DNA. That's the main DNA that's in the middle, right? And then they had these little itty bitty cute guys called plasmids, right? These little circles of DNA that they can swap back and forth. Remember that discussion? So as we take a look at the chloroplast DNA, what we notice is that they are little circles. And they reminded us of something. They reminded us of bacterial plasmids. So what you were missing was the nucleoid DNA, the main DNA genome of a bacteria, but you still have these like little plasmid, these little rings of DNA. So it kind of is about the same size. To a bacterium. So for instance, a bacteria is roughly between 1 to 1.5 microns in length and about 0.5 micron in width. Guess what the dimensions of a chloroplast and mitochondria are? Similar. So it's got 1 to 1.5 in length, 0.5 in width. So let's add this up. You've got a plasmid looking or bacterial looking plasmid in you. You're roughly the same dimensions as a bacteria. So the question is, were you at one point a free living bacteria, right? That gets us to an evolutionary idea called the endosymbiosis theory. So endo means inside, and a symbiosis is a mutually beneficial relationship between two organisms. So the idea, again, this kind of goes back to evolutionary modeling, right? Now remember, according to, basically when we're talking about this, we're talking about events that probably happened maybe 3-4 billion years ago, right? And remember our discussion at the beginning, you have to be careful the further back you go, the harder it is to know for certain and have certainty about these ideas. And so we see that reflected in this one. But one of the interesting branches of, I guess you could call it theoretical evolution, was trying to figure out how all this kind of came together, right? How this all came to be. Like, for instance, how did we get a very complicated eukaryote, right? With all these organelles in it, like where did all these things come from? How did they actually come together? And the endosymbiosis theory tries to sort of explain at least a little bit of that, how that could have happened through a model. And there's different versions of this, of how this could have happened. So for instance, one of the things that they suggest is that basically what you had back in the day, and there's, we don't know, there's like probably at least a half dozen versions of this idea out there. So some say that it was an ancient eukaryote that basically gobbled up a protobacterium, right, an old ancient bacteria. And then when it gobbled it up, because if you recall, one of the things that happens when you engulf something, we saw this, right, is you surround it, right? And then when you internalize it, it has now two membranes, right? It's got its own membrane and the membrane that you just wrapped around it when you engulfed it. So when you're looking at an engulfing theory, it seems to be consistent with the double membrane organelles that we see in the cell. So the question is, where does the double membrane come from? The answer is perhaps by engulfing, right? Because basically that would have given you a double membrane system. And if this guy was making a bunch of ATP, and that's what he did for a living, and he did it quite well, you could imagine this ancient eukaryote basically pausing and thinking, you know what? You're making a lot of energy, and I'm using that energy. You're actually more useful to me alive than if I were to chop you up and digest you and eat you, right? So why don't I not eat you? Why don't you hang out with me and make all the ATP you want? I'll give you whatever you want. I'll protect you. I'll give you any kind of molecules you want, any kind of food you want. Just name it. I'll give it to you. But the deal is you make ATP for me and I will protect you. That's an endosymbiotic relationship. And so obviously over time, the mitochondria or this protobacterium, they basically let some things go, right? So for instance, it let a lot of stuff go, but it still held on to a few things. Like for instance, it's a little plasmid, and it explains where that double membrane came from. Okay. Yes. Engulfing. Yep. And the idea is for a plant, you did the same thing. Only in this case, it was a cyanobacteria, photosynthetic bacteria, like Glyocapsa and Oscillatoria, right? Those are our two cyanobacteria that we saw in lab 6. And so in this case, if you engulfed cyanobacterium, then basically you had what was the chloroplast, right? So animals basically engulfed this protobacterium, this mitochondria-like protobacterium, and plants did a second engulfing and engulfed a cyanobacterium, and that basically became the chloroplast. Well, the nucleus also has double membrane system, doesn't it? So the idea there, and this was a little bit of an addition later on, was that maybe the origin of a nucleus is that there's some sort of an archaean, an ancient archaean that was actually engulfed, and that became the nucleus, which is fascinating, right? That would suggest that most of your genome is archaea, which would explain some things that we see, right? It would explain the reason why archaea have a similar DNA replication system to us. It would also explain the reason why they have a similar protein synthesis system to us. And so that's kind of an interesting thing. Now, nobody knows for sure, right? I mean, these are things that when you do modeling like this, what you rest on is what's called explanatory power, right? So the more that your model is able to explain current phenomena, then the stronger, more robust it is. The more of those you can explain, the more likely you are to be correct. Okay. However, but you never know for sure, right? So it could be there's a half dozen of these things out there. It could be that none of them are right, and that it all came together in some other way that we haven't thought of yet, right? Which is always the big caveat with these sorts of things. But it's fascinating. It makes for interesting reading, and it's a lot of fun. So that's basically the end of the energetic section. Now let's get into the cytoskeleton section, right? So basically when you're talking about the cytoskeleton, you're talking about the support for the cell, right? This is your structural framework. And so it's a network of fibrous proteins, and largely their job is to support and create shape for the cell. So there's a lot of this that basically is static. Some is static, and some is dynamic. So some portions of the cytoskeleton is designed to be dynamic, and some portions of the cytoskeleton are designed to be very static and stationary. Okay, and we'll take a look at which pieces are which.
The dynamic is when they are able to move and change form.
Correct. So like, for instance, if you can build it out and break it down like a Lego set, then that's dynamic, right? So you can build some sort of a scaffolding structure and you can tear it all down, and you can rebuild something else and you tear it all down, rebuild something else, tear it all down. That's a dynamic system. Okay. Static basically means you build it and you're done. You walk away, you're not going to change it, right? Because if you were to deconstruct it, you would have catastrophic results, right? So we'll kind of see the oscillation of those two going back and forth. The first thing we need to do, I know we've already been kind of introduced to these guys a little bit, right? Because we outlined in our outline the three major fibers of the cytoskeleton. We've already been actually introduced to the third one, haven't we? The intermediate filaments. So on my outline, I listed them according to size. Right here, they're not listed according to size. They're listed according to whatever. I don't know what they're listed according to, but not size, right? But we've already seen intermediate filaments, an example of that, right, in the nuclear lamina. So we'll talk a little bit about that here in just a second. But the first one we see, and the smallest, is going to be what's called microfilaments. These guys are going to be made of actin. So a single subunit of actin, which kind of binds to itself and forms this chain. That's the reason why it's called an actin filament. But it doesn't just have one chain of actin. It's actually got two chains of actin that kind of intertwine with each other. And that kind of forms your little actin filament. It's fairly small. This is the smallest of the three. And it is the culprit behind three major types of activity. So contraction, for instance, like muscular contraction, it's a major protein fiber structure in protein. And you'll get a lot of that when you get to AMP1, for those of you going into AMP1. And then there's crawling, which is like a local movement. This is like moving, like a cell moving by oozing along. So that's what's crawling. And then, of course, pinching. So when you actually kind of like create like little fleshy fingers and it pinch at something. So there's three major types of movement. We're not going to talk about contraction in this semester because AMP covers contraction. We're not going to really talk a whole lot about pinching because that goes into cell biology. But crawling, we will take a look at because that's actually a fairly important mechanism. We'll take a look at that with cell movement and locomotion in just a few slides. So that's an important component of your cytoskeleton. Then you have microtubules. Microtubules are kind of like the big sexy, if you will, right? This is the one that everybody wants to study. Right, this is like if you talk to a biochemist out there studying cytoskeleton, most of them are studying microtubules. Not a lot of actin research, not a lot of research, but on research. And microtubules basically are long cylinders made of two different types of protein called tubulin. You have an alpha tubulin and you have a beta tubulin, and they form what's called a heterodimer. So they form a pair like Batman and Robin. And then the way you build your microtubules is you basically just stack your tubulin dimers like bricks, and you just kind of form what basically becomes like a long cylinder. And so that's basically your microtubule. And the reason why microtubules are important is because they facilitate the movement of the cells and stuff within the cells. Microtubules are incredibly important when you're talking about moving a stuff around in the cells. And we'll talk very at length about microtubules. Actually, most of our cytoskeleton discussions will be about microtubules. Okay, so I don't want to let the cat out of the bag too much right now. Now, intermediate filaments are basically kind of like, these two are dynamic. That means you can build them up and break them down, build them up and break them down. So microtubules and actin are designed to sort of build out, break down, build out, break down. So they're dynamic. Intermediate filaments are not. These guys are static. You build them once and they're there. So this basically creates the scaffold of the cell. And so it's made of different types of proteins. So for instance, we saw laminin making up the nuclear lamina, which is a long fiber of laminin, right, in a chain. But we also have keratin making up hair and nails. Notice different protein, same basic idea. You have some sort of a structural protein, could be different things, and they basically make this long sort of filamentous fiber that then becomes structural. Now, the important thing about intermediate filaments is that they are stable. They don't get broken down. Once you build them, this is your scaffold. So remember my analogy of the tent frame holding up the tent fabric. The frame itself, which is not designed to move at all, is like the intermediate filaments. It basically holds everything up. So you don't change the tent frame. Okay. Now, this image I like a lot because it puts the three fibers into its context in the cell. So for instance, the first one you're going to see here is the actin fiber. Notice you have two chains twisted together, but both of those chains together only come out to be about 7 nanometers in diameter. So this is the reason why they are the smallest. But you can see these guys out here sort of at the periphery of the cell in various amounts. So you can see these little 3 actin filaments at the periphery of the cell. The next big one is going to be the microtubules. So these guys are the big ones. So this is 25 nanometers, and you can see how you have your tubulin subunits. So they're light and dark green to show you the different types of tubulin, alpha and beta tubulin, that are kind of stacked up. And you can see it's a long cylinder, like a big straw, that's basically sort of scattered throughout the cell. So you can see one here, there's another one here, there's a microtubule here, there's one here, there's one up there, there's one down here. So you can see they're kind of scattered all throughout the cell. And the important thing about microtubules is that unlike the other fibers, they have a polarity to them. That is to say that they have what's called a plus end and a negative end. Now the plus end is important because this is the growing end. So what if you wanted to extend this microtubule, grow it out? It's easy. All you do is you stack tubulin to the end of the plus end and you can grow it out in that direction. What if you want to contract it, pull it all back? Then you just unstack the tubulin and it basically shrinks. So microtubules, unlike the other fibers, have the capacity to extend in length as you stack tubulin on the plus end and to contract backwards or to shorten as you take tubulin off. Okay, so it's a dynamic process and they're very, very dynamic in nature. Now, the intermediate filaments, which are somewhere in between in size, right? So they're about 10-ish or so, and they varies depending on the protein you're using, is actually kind of like a cable of cables. So if you've ever seen a suspension bridge cable, like a Golden Gate Bridge or Bay Bridge or like the Brooklyn Bridge, right? So one of these big wires that basically holds the bridge up. If you take a look at a cross section of that big gigantic cable, it's a cable of cables. So inside that are going to be smaller cables that forms that cable. And inside those are going to be smaller cables. Yes. They can. They have a little bit of flex. That way you can, the expansion, contraction of the bridge and the movement of the bridge, you can kind of, that's one of the nice things about it. But these smaller cables have even smaller pieces in it. So it's like a cable of cables, like nested cables. And the same thing is true here. So here you can see kind of like your big suspension bridge cable. And then within that, you can see smaller cables that are then also made of these little fibers of protein, whatever that protein is. Laminin, if you're nuclear lamina, keratin, if you're a keratinous sort of a thing. So those are intermediate filaments. But if you take a look at the intermediate filaments here, you can see how the intermediate filaments actually are spread throughout the cell. And this is one of the reasons why I like this particular image, because there's not a lot of textbooks that do a good enough job showing you how all these pieces sort of lay out in the cell. So here you can see the intermediate filaments basically form this sort of scaffold-like structure. That's basically all these tan pieces. And it kind of holds the shape of the cell. Everything, it kind of like just basically holds up the basic structure of the cell. And it doesn't change. It always stays there. So once you build this iron framework, if you will, it just stays put, right? So it basically holds the cell up so that you can do stuff inside the cell. And not only that, but it also attaches stuff to it. So not only does it hold up the cell membrane and give the cell shape so it doesn't collapse like a deflating balloon, but it also attaches organelles to the cytoskeleton. So for instance, in your rough ER, what you have here it basically is tethered in a single place by these intermediate filament cytoskeletal connections. What that does is it basically means that your organelles are not free and floating around inside the cell. They're anchored in position to the cytoskeleton. This is important because remember when we talked about the endomembrane system, where we talked about the vesicle going from the rough ER to the Golgi? Well, if the Golgi wasn't anchored in place, then it'd be like trying to shoot a moving target, right? So your vesicle will be like, okay, wait a minute, where's the Golgi today? So yesterday it was over there. Today it kind of floated over here. So now I got to figure out where to find it, right? So that is hugely inefficient, and that is not good biology. So what the cell does, say, listen, I want the Golgi to be right there every single day. And vesicles, I want you to move to the Golgi along a definable pathway with a definable destination, okay? So it keeps everything in place so that you can actually get your work done inside the cell. So incredibly important. Now let's take a look and talk a little bit about, that's basically probably the last we're gonna talk about intermediate filaments. So we're gonna talk a little bit about microtubules, get some microtubule stuff set up. We're gonna talk parenthetically about actin filaments, gonna talk a little bit about the crawling mechanism, and then we're gonna basically move and finish up with the microtubules. So when you take a look at microtubules, you have these plus ends, right? So you have these growing ends, and you have the ability to sort of build them by just stacking tubules. But here's the question: how do you get it started? Where in the cell do you start building microtubule? Do you build the microtubule by starting to stack your tubulin out of the periphery of the cell? Do you do it by moving in toward the center of the cell? Where in the cell do you start, right? Where's your start point? How do you know where to start building your microtubule? And so the important thing that we see in order to get microtubules going is we see a structure in all cells called a microtubule organizing center. This is basically a nucleation point for microtubule construction. So an MOC, a MOC, right? A microtubule organizing center. Now, as it turns out, in animals, their microtubule organizing center is the centrosome. It's a specific organelle in the cell that does this particular job. Now, the centrosome itself is composed of two drums of microtubules that are perpendicular to each other, called centrioles. So each one of these drums is a centriole. Both of these guys together is a centrosome. Okay. Now, these little drums basically are a set of microtubules that allows you a starting point, a nucleation point, where you can start building your microtubule network. Now, this is the MOC in animals, but not in plants. In plants, there are no centrioles, but it does have a MOC. Yeah, Chris. Yes, they're right next to each other. They're like this, like a T, always. Now, what holds them together? I'm not sure. That would get into the ultrastructure of like a centriole. But there's probably some mechanism or some structure that holds them together so they don't separate out. I just haven't gotten that detailed. That's like like when you're getting deeper into cell biology, you start looking at more of the fine structure of the different organelles. That's usually where you get that one. So one of the things, though, that you need to be aware of is in some sources, in some textbooks, and none of the textbooks have this now because they've written it out because it's obviously a mistake. Actually, it's an incorrect correction is what it is. Is there have been some books that basically will say crazy things like, like in a chapter like this, where you have some sort of an organelle, a common set of questions would be like, okay, I'm pointing to an organelle. What is this? And what does it do? Right. So that's kind of that's a common question that you see in the chapter like this. And a lot of textbooks, you get to the centrosome and they say like, okay, what is it? Oh, this is a centrosome. What does it do? And then the book would say, we don't know. I'm like, no, we do know what a centrosome does. And the reason why they say we don't know is because they say plants don't have centrosomes. So because plants don't have centrosomes, we don't know what centrosomes do. That was their reasoning, which is wrong because we know exactly what centrosomes do. Centrosomes are the microtubule organizing center in animals. And we know exactly what a microtubule organizing center does. It's a nucleation spot for microtubule development in animals. Now, do plants have centrosomes? No, but that doesn't mean we don't know what centrosomes do. What it means is we don't know what plants use as a microtubule organizing center if they don't have centrosomes. But they do have one. They have to, because they organize microtubules too, just like us, right? They just do it differently. So it's not true that we don't know what the centrosome is. And I caution you, because as you guys are sort of out there scavenging and scouring the planet for, studying for the exam and things like that, if you may run into that. Just know that they're wrong, because we know full well what centrosomes do, quite clearly. So this is basically your centriole. So you can see that your centrioles actually have a very definable structure. So you can see it's basically just a network of microtubules, like little short microtubules. You have 3 different microtubules that are essentially connected together, and you have 9 of these, right? So you have 1, 2, 3, 4, 5, 6, 7, 8, 9. So there's 9 triplets around a hollow core. So you notice you have nothing here in the middle. It's just like an empty straw. And you have these guys in perpendicular. So here you can see the individual, there's proteins that are attaching these individual triplets to each other. So it's a nice little ring. But what this does for you then is that when you want to build microtubules, you can pretty much build these in all 3 dimensions, right? So you can basically start building off your microtubules in all directions, however you want, as many as you wish. And they're going basically everywhere. You want to build in this direction? Great, let's build off of this one. And build straight, build over here. So you can see how very quickly I can basically start to build a microtubule network that pretty much crisscrosses the entire cell, goes anywhere I want it to go. Okay. So that's kind of what your microtubule network looks like. And it's all basically starting with your centrosome, right? That nucleation spot. All you have to do is start stacking tubulin. That's all you do. Just start stacking tubulin on the end and grow out your microtubule. Okay, so let's put this to work now. Let's put our cytoskeleton to work in terms of its major innovation, which is movement. Yeah, Christopher, go ahead.
Is there a negative byproduct or
No, like shortening them? No, you do that as a matter of course. So it's a normal operation for the cell. They're constantly extending them out and shortening them up. So there's no harm, no foul. It's not like a pathological sort of a thing. It's completely normal. It's kind of like cutting your hair is like a normal thing. It's not like a sign of disease or devastation, right? So it's just you're cutting your hair. That's all it is, right? It's just kind of normal business. So we're going to be taking a look at 2 different types of movement here. The first one involves microtubules, and we'll take a look at this one a little later. But before we get to that one, what I want to do is I want to take a look at the actin-based type of movement. And in particular, I want to take a look at crawling, right? Which is basically just movement across local distances, small local distances. This is not your strategy to move far. And in order to do this, what I want to do is I want to add a slide and I want to sketch it out. Because we do have cells in our body that actually do this type of movement. Our white blood cells will do this. Our immune cells will do this. As they're moving through your tissues, they kind of move. This is the mechanism for moving by oozing. It's called amoeboid movement, because amoeba actually do the same type of movement. And this is the same mechanism that your white blood cells use to move around your body and to engulf cells and things like that. So let's imagine we have an amoeba. So there's our nucleus. Now, one of the things you need to understand is that just underneath the surface of the membrane is a lattice work, like a scaffold of actin, like an actin network. And this is called cortical actin. And it's solid. It's a solid matrix, and it goes all the way throughout the periphery of your cell. Now, also in your cell, at least in amoeba, is more actin, but there's actin fibers that are just kind of like floating around in your cytoplasm. So this is basically cytoplasmic actin, and it's kind of like a gel fluid. So it's got a flow to it. Now, let's imagine that this particular cell, which in this case an amoeba or a white blood cell, wanted to move over to here, this direction. The way it does that by oozing is that the first thing it does is it breaks down the cortical actin in the region that it wants to go. So now it exposes naked membrane. And then what it does is it contracts, it kind of squeezes like you're squeezing a balloon to make it bulge. And when it squeezes, it forces the fluid against that open membrane, and it basically puckers that membrane out like that. And inside is all that fluid flowing actin that's pushing that little piece of membrane out. Just like if you were to squeeze a balloon and you cause it to bulge, right? That air is going to be pushing against the balloon and making it bulge. This is what's called a pseudopod, false foot, what that means. And then you anchor that to where you want to go. And then if you like that and you want to stay there for a while, then what you do is you reassemble your actin into its cortical network. That solidifies that foot. That foot's not going to move then once you get that cortical actin in there. And then if you want to move the rest of your body over there, you just break down your cortical actin and you just kind of use the fluid to kind of pull yourself along, and you rebuild your cortical actin. So as amoeba move and as your white blood cells move, they're constantly doing this dynamic kind of breaking down your cortical actin, pushing against your membrane, moving somewhere, rebuilding your cortical actin to keep you there, right? And then kind of breaking it back down again to go somewhere else and that sort of thing, right? So you're kind of constantly doing this back and forth with your actin. That's amoeboid movement. That's crawling. So you're going short distances. We're talking tiny distances, right? You're not moving very far, very fast. Yeah. Because they're what? No, the reason why they do that is because it's easier. That's the only way a cell can move. There's really not a mechanism in your body for cells to move. The only way they can move is by just kind of like tumbling around, oozing around. And white blood cells have to patrol the tissue. So they have to be able to ooze around and move around. Other cells, because they're connected with cell connections, which is one of the last discussions we have in this chapter, can't do that because they're battened and fastened in place. The only cells that move don't actually move. It's our blood that moves them. So they're borne along by the fluid of our blood. So that's the reason why they have to move. Okay, so let's go ahead and take a look at what if you have to do longer distances? Well, then you have to basically have a different system, and we do. So for longer distances of movement, we use what's called flagella and cilia. So for instance, a flagella is kind of a long whip-like structure. Cilia basically are kind of short hair-like structures, but they have the same microtubule structure, what's called the 9+2 arrangement of microtubules. We're going to take a look at that structure here in just a second. And we're going to see how you can actually get movement from the mechanism of the flagellum. It's very different in structure than the prokaryote, right? So it's not the same as the prokaryote. And so we're going to take a look at that. But before we take a look at that movement of the flagella and how you can move things along, we need to actually talk about a couple of players, right? So first of all, we need to talk a little bit about motor proteins. So when you're taking a look at moving stuff around the cell, right? So then the thing about movement is none of the movement is random, right? So when we take a look at vesicles in the cell, it looks like it's just sort of randomly floating from the rough ER to the Golgi, but that is not the case, right? What we don't see is we don't see the little motor protein underneath the vesicle that's actually carrying that vesicle along to its destination. Remember, in the cell, nothing is random, right? Everything is very intentional, everything is organized, and everything is laid out. You don't let things drift. Why? Because drifting is random, and the efficiency of drifting is very low. And you don't have time or money for inefficiency, which is the reason why everything in the cell is intentional, okay? And so what is the intention? How do you get the vesicle from the rough ER to the Golgi? Well, it's simply basically a commute. So just the same way that you have an analogy, just the same way that you got here to campus, right? A lot of you guys drove here perhaps. And if you drove here, there's a couple of things you need in order to actually drive here, right? First of all, you need a pathway to get here, yes? You need a road. In the cell, those roads that take you from one place to another to your destination are the microtubules. So these are your roads that take you to where you need to go. In this case, if you're needing to go from your rough ER to your Golgi apparatus, you have a microtubule that takes you there. There's a connection there. As a matter of fact, not only that, but if there's a destination that you need to get to that doesn't have a road, what do you end up doing? You create a road, right? So road building is dynamic, isn't it? You have to go somewhere new, create a road. If you have a destination or a road going somewhere that you don't ever go to anymore, what do? Break it down, right? So you don't need a road there anymore because nobody goes there anymore. So road building is dynamic. We're constantly building them out. We're constantly retracting them. We're constantly rerouting them. And so the same thing is true for the microtubules. The microtubules are the roads. These are the pathways through the cell that determine where you're going. Now, what else did you need in order to get here? Well, you needed a car, right? Now, a car is the motor protein. So a car has a couple of domains, just like a motor protein does. The first domain that it has is what's called the walking domain. This is the domain that actually makes contact with the road and moves it along. This would be analogous to what? This would be the wheels. Your wheels are on the road, right? Moving along. But guess what? As you're walking down the microtubule, you don't get that for free, do you? You need what? Energy, right? That's your ATP. That's going to give you your energy. So this would be your gas plus your motor, because your motor needs the gas, right? So that is what's called your motor domain, right? That's basically the motor domain of the car. But then you also have another part of the car, don't you? You have your cargo binding domain, right? Where's that in the car? The cabin, right? It's where you sit. That's your cargo binding domain. So just the same thing. Motor proteins have all the same things that your car has. It has a walking domain, wheels to contact the road so it can move along the road, which is the microtubule. It has a motor domain that requires energy, right? Just like there's a motor domain associated with the motor proteins that requires ATP in order to do this. And there's a cargo binding domain. The cabin of the car is a cargo binding domain, just like there's a cargo binding domain associated with the motor protein. So you have all of these. And then, of course, your cargo binding protein will bind to your cargo. In this case, it's a vesicle. And if you have gas, a motor, and your cargo, then your wheels will roll down the pathway, wherever the pathway takes you, right? In this case, you're rolling down the microtubule. Good. Different motor proteins? Yes, there are many different types of motor proteins, and they do have different speeds, but they have different functions as well. So they do different things. Just like not everything is a car that's taking you to school, right? Some things on the road are buses, some are trucks that are hauling stuff somewhere else. So there's similarities, but they have different tasks, right? So now that we have our mover, right, our people mover, if you will, now we can start taking a closer look at how we can put all this together. So we've got basically a flagellum, we've got a motor protein that walks along the microtubule. So we got that piece. In this particular case for flagellar motion, we're going to be focusing on the dynein motor protein specifically. And then what we're going to do is we're going to take a look at the movement mechanism of the flagella. So first of all, we got to start off with the structure, right? We haven't talked a little bit about that 9+2 structure. We need to in order to understand how this works. So first of all, let's start off at its base, right? Just like you did with the centrioles, building your microtubule network from your centriole base, you also have to have a base to build from for your flagella. And that flagella base is what's called the basal body. That's actually in the plasma membrane. And if you take a look at the construction of the basal body, it should look very similar to you. Notice you have what? triplets surrounding a hollow core. That's actually not a microtubule, that's just like protein stuff in there, right? But there's a hollow core, very similar in structure to your centrioles. Now from this, you're going to build out and extend your microtubules. And then as you kind of get to the mature area of your flagella, we're going to take another cross section so you can see what the 9+2 structure looks like. So once you actually get your flagella kind of built out a little bit, what you're going to notice is, first of all, you have now not triplets, but doublets. And you have 9 of them, right? You have 1, 2, 3, 4, 5, 6, 7, 8, 9. So that's what the 9 is. So you have 9 microtubule doublets around a core of 2. Not connected. So that's your 9+2 structure. A ring of 9 doublets around a core of 2. Okay. But we're not done, because we have other proteins now in this mature flagellum. Notice these little cloudy pieces right there. There's one right there, 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 there's one right there. Those are what's called radial spoke proteins. They're connecting the doublets to the central hub. So what is it starting to look like now? You've got this 9 doublet ring around a central hub connected by spoke proteins. It's looking like a bike wheel, right? Which is important, because let's take a look at just the basic physics of a bike wheel. A bike wheel is an interconnected system, right? You've got a rim that's connected to a hub by spoke proteins. So anything you do to the rim is going to spin the hub. Anything you do to the hub is going to spin the rim. But not only that, but if I pull on the rim on the right side and I pull it down, what's going to happen on the left side? It goes which direction? It'll go up, right? So pull this side down, this side goes up. Why? Because it's all interconnected. Because the force that you're putting on this side is radiating throughout those spoke proteins to the opposite side of the rim and forcing that to move over. Okay, that's an important idea to keep in mind as we look at flagellar motion. There's one more player here that we need to introduce. We haven't introduced, we just need to highlight it. Notice these little blobs right here. Little Mickey Mouse ears on these doublets. Those are dynein. That's your motor protein. And so when you take a look at your microtubule doublets with these dynein Mickey Mouse ears on it, all interconnected to the radial spoke, by radial spoke proteins to the hub, what you have is a network of microtubules that are all interconnected with each other. So what you do with one is going to be radiating and felt throughout the entire system. That's important, because let's take a look at it. So here is my microtubule doublet. Here's an adjacent one. So basically the way this is going to work is your dyneins, the cargo domain is bound to the microtubule doublet. So you got the little walker domain sticking out, the little leg sticking out like this. And the same thing over here. Now, the way that flagella move, yes. Well, the long cylinder is going to be felt all the way throughout the entire flagellum. Okay. So basically the way flagella move is by alternating the walking of the dyneins. So notice if these are my 2 microtubule doublets and you have my little dynein leg sticking out, the way you walk is that one doublet, the dyneins will grab onto adjacent doublet and start walking. And once they walk, what's going to happen is they're going to pull the other doublet down. So the reason, as this is going down, because everything is interconnected, this side is going to go up. But remember, it's all interconnected. So it's going to go up and it's going to get pulled over like that. And it's going to bend, just like the bike wheel. But then what happens is those dining feet let go. And then when that happens, so you go from straight to a leftward bend. And then when they let go, they go back to straight. And then what happens is the other one does it. It attaches and then it starts to walk and it pulls this guy down, bending this whole system over to the opposite side. So then you get a rightward bend. And then when they let go, it snaps back straight. And then you alternate back and forth, left, right, left, right. And so what you get if you speed it up is it starts off straight. Then you get the first walking, which bends it to the left. It lets go, snap straight. Then you get the second walking, which bends it to the right. Let's go, snap straight. If you speed that up and alternate it, you get bend, snap, 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 bend, snap. And if you're a flagellum on a cell, you're basically paddling away and you're swimming. Okay. Or if you're cilia, you're basically doing this. Kind of like kelp, bending back and forth and back and forth. Okay. And so in us, we have one flagellated cell, and that's sperm. And that's what creates sperm motility, right? Is flagellar motion. And so it's significant enough. And actually, I mean, there's a little bit of distance between the point of deposition and where the egg actually is in the fallopian tube. So the sperm actually has to swim a little ways, right? It has to go through the female reproductive tract, it has to go up through the fallopian tube, it has to actually find the egg and then fertilize it. So it's got a little bit of work to do, right? So the sperm have to be motile. And this is what gives us some motility. And we don't have a whole lot of ciliated cells either, but we do have one primary spot of ciliated cells, and that's in our trachea, right? In our respiratory system, we have cells that are ciliated, that are constantly waving back and forth, and they're secreting mucus. So what you have is kind of like this gummy mucus marsh that's kind of bending back and forth with the wave of cilia. And the reason why that's there is because when you breathe in dust particles, those dust particles get trapped in the mucus, and the cilia just basically sweep that up your respiratory tract into your cough reflex, and then you cough it out. Okay. This is one of the reasons why smokers have that really crackly smoker's cough, is because they're constantly bathing their respiratory tract in smoke particles. It's getting like lots of mucus is getting caught in those smoke particles getting caught in the mucus, and it's like they're constantly sweeping it up. Which is why it sounds like they're hacking up half a lung, like all the time. That's because of the smoke particles. Now, this is kind of like a little bit of a parentheses, just to kind of like tie things up for plants. Got to give plants a little bit of love too, right? The cell wall is obviously made of cellulose, but there's actually more than just like a bunch of cellulose. There's actually layers to the cell wall. And we're not going to get too much into this, but here you can see you've got cell number one. And as you start to move through the cytoplasm, you're going to first run into the plasma membrane, you can see here. The first layer you run into, which is the dominant layer we think of, is what's called the secondary wall. So this is your main sort of cellulose layer. And then outside of that, you're going to see the primary layer, which is another layer beyond that. And then in the middle is what's called the middle lamella. This is the gap between plant cells, and it's kind of like full of sort of like a goo, if you will, that sort of glues cells together. And you kind of reverse the process, right? So you kind of have your primary cell wall here, and then your secondary, and then your plasma membrane for the next cell. Okay, so there's actually different layers of the cell wall. If we were in a plant physiology class, we'd get into a little bit more of the fine structure of plant cell walls, but it's not necessary. It's sufficient just to know that there's, a little bit more to a plant cell wall than just a bunch of cellulose piled up there. Okay, so that's the end of our big four. Now what we have is a couple of topics that that are important, but there's about 2 or 3 topics that toward this end that kind of are important. The first one is the extracellular matrix. So the question about the extracellular matrix is, what exactly is it? Well, basically what the extracellular matrix is a secreted environment where each cell will actually secrete stuff to the outside of the cell itself. And it'll create this kind of surroundings, if you will, this matrix around it, called the extracellular matrix. And typically it's a mixture of stuff. It's a mixture of something called ground substance, which typically has lots of slippery molecules in it, a lot of carbohydrates in there, protein stuff in there. So lots of molecules that are like lubricating. This ground substance can vary between a very fluidy substance to a more gel-like substance. There's different things you can do. Different cells can sort of secrete things very differently out there, depending on what kind of a cell they are. Most all of them will also dump out their protein fibers. And one of those protein fibers that we have is collagen, right? So collagen is basically a tough structural protein. And it's the dominant protein in your connective tissue. So if you want to know what pure collagen looks like, as close to pure collagen as you get, grab a hold of your Achilles tendon. That's nearly pure collagen. It's like a big collagenous rope. So it's really tough and really fibrous. And there's obviously other proteins that you can stick out there. And the important thing about this is the extracellular matrix for some cells is protective. For others, it's not just protective, but can be also very functional. So for instance, what we think of as the tissues in AMP, a lot of times is actually the extracellular matrix. That's what it is, like bone, for instance. Most of your bony tissue is actually extracellular matrix. Same thing with cartilage, same thing with connective tissue. And so depending on who you are as a cell will dictate what your extracellular matrix looks like. An extracellular matrix is very specific to the cell. As a matter of fact, I like to think of the extracellular matrix kind of like a bird's nest, right? If you've ever run into an ornithologist, a good ornithologist can actually do a bird identification just by looking at a bird's nest, because birds have a very species-specific way of building their nests. Some are weavers, some are mudders, some are burrowers, some are trash collectors, right? They get that, piece of gum wrapper in there that's all shiny and silver, and that's apparently supposed to be attractive to the ladies, apparently, right? Just imagine that's like, hey, got me a gum wrapper, right? So that's apparently a thing. Don't try that, by the way. That'll get you pepper sprayed in most establishments. Just saying. But it gives you a sense of context, right? Not only that, but it gives you a sense of anchorage. So let's face it, we just talked a little bit about motility, right? So cell motility. And we also mentioned that most cells in your body are not, because they're fastened together as sheets of tissue. So generally speaking, for most cells in your body, floating around is a bad idea, right? You do not want to just float around randomly, because you can't get the kind of organization you need to do your business. So anchorage is critical, is absolutely critical. Which is one of the reasons why one of the most dominant tissues in your body is connective tissue, because it is like support structure for your body, right? To give your body anchorage to something solid. The same thing is true for the cell. So you can think of the extracellular matrix as a way to anchor the cell and stabilize it so that it can basically do its business instead of being buffeted about all the time and having to worry about whether or not it's going to be in any one place at a time. And so in order to do this, the cell has an actually clever system. What it does is it basically takes a protein called integrin and it inserts it into the cell membrane, and then it basically uses that to tether itself to its extracellular matrix. And this is kind of what it looks like. So here you can see, here's your integrin molecule. You can see how it's inserted into the membrane. And then on one side, on the cytoplasmic side, it's actually tethered to the actin. This is your cortical actin. So it's anchored to your cytoskeleton. Then on the cellular side of it, you have your extracellular matrix out here, and you have all sorts of different fibers out here, right? So you have your tough collagenous fiber here. You have another protein fiber here, elastin. This one is for flexibility. You need a little bit of flex. You have other molecules like this, proteoglycans. So this would be part of your ground substance, and all kinds of stuff going on out there. But remember, your collagen is your tough fiber, right? That's your structural fiber. So what your cell does is it basically creates another protein called fibronectin that's designed to latch onto your collagen. What this does is it stabilizes you. So it anchors your cytoskeleton to your external cellular matrix. Think about it. Let's imagine that I'm walking along and I get a little unsteady, right? So if I'm getting a little dizzy and I'm about ready to go down, what's the first thing you always automatically try to do? Grab something, right? So what are you doing? You're doing exactly what the cell is doing here. In one sense, I've got a grabber, right? This would be like my integrin fibronectin network, right? My arm. But my grabber is connected to what? My skeleton, right? My structural component of my body. And so when I basically grab onto the environment like this, I'm grabbing onto what? My extracellular matrix. I built this, just like the cell built its own extracellular matrix. So this building wouldn't be here if I didn't build it. This is mine. So when I'm trying to steady myself, I can grab onto my own extracellular matrix. By the way, do I grab onto the chair? No, I built this one too, right? So that's a bad idea. I don't want to grab onto something that's moving. That's why I'm not grabbing onto elastin, right? That's flexible. So what I do, I grab onto something tough and structural. I grab onto the collagen because I know this is anchored to a strong wall. I anchored this down, right? So I steady myself so I can basically have anchorage. So that way I have a point of reference, and that's always an important thing for a cell, anchorages. So once we actually know how to anchor cells, once cells actually make their extracellular matrix, now let's go ahead and start putting cells together to actually form tissues, because that's what we need to do. We do this with cell connections, right? And so first of all, when you take a look at the surface of the cells, there's different things out there. The first thing you need to have out there is cell identity markers. And there's two different major types of cell identity markers, glycolipids and also glycoproteins. These both are involved in cell sorting and identification. So think about it. You want to make liver tissue. Liver tissue is made out of liver cells, right? So the question is, how does one liver cell identify another liver cell to know that, hey, we need to batten together and form team liver, right? That means that there has to be some sort of a recognition strategy. So your glycolipids and glycoproteins profile on your surface looks to other cells like, hey, I'm a liver cell. So when you see that profile of glycolipids and glycoproteins, it's like a name tag. Hi, my name is Mark. I'm a liver cell. Oh, hey, my name is Mark too. I'm a liver cell too. Okay, let's go ahead and form connections now so we can form team liver, right? Notice I said two different things. Hi, my name is Mark. That's one thing. And I'm a liver cell. That's the second thing, right? So the identification of I'm a liver cell is driven by glycolipids and glycoproteins. A type of glycoprotein, a specific type, is what's called the major histocompatibility complex, the MHC. This is your self tag. This is the hi, my name is Mark. Every single one of your cells in your body has your name tag on it. Your immune system grows up recognizing your name tag. So wherever it sees your name tag on a cell, it permits it to live. If it does not see your name tag on a cell, then swift death will ensue. So for instance, my liver cells say, hi, my name is Mark, and I'm a liver cell. But if I get a liver transplant, then all of a sudden that changes to, hi, my name is Bob, and I'm a liver cell. The liver cell's fine because that's going to save your life, right? But the Bob part is unacceptable. Your immune system sees Bob inside you and goes on the attack. This is the basis of organ rejection. It's the reason why if you're an organ transplant patient, you're basically going to have your doctor is going to be part of the family. And you're going to be on a myriad of medications. Many of them are immune adjusters to try to keep your immune system from completely destroying and rejecting your organ. So that's just the thing. So for you table people, this is your tables. So you can see basically what's out there for cell identity. You've got surface markers out there. You've got different types of connectors that actually connect cells. So you have 3 different types. You have a really tight connection between cells, a fairly loose connection that goes for communication, and you have something in between, right? Notice there's a trade-off here. whenever you're connecting cells. You can either pick communication versus connection. But you can't have both, right? Your tight junctions are going to be connection at the cost of communication. Your communication junctions are going to be communication at the cost of connection. And your adhesive junctions are somewhere in between, a balance between the two. So let's take a look. Now that you've identified each other as marked cells and you've identified each other as liver cells, let's go ahead and start putting ourselves together. We're going to start off with the adhesive junctions. So the adhesive junctions basically are they're kind of a combination of the two. So this is a balance. Of communication versus connection. So this is one different type. There's different types of adhesive junctions. There's what's called adherins, there's desmosomes and hemidesmosomes, and there's a whole bunch of others. The example that we have here is a type of adherin called cadherin. This means calcium dependent adherin. And it has a similar type of a theme that we saw before, right? So first of all, one part of this complex is anchored to your cortical actin, right? That's your cytoskeleton stabilizer. And then it'll have an extracellular piece to it, a receptor that kind of sticks out. And then an adjacent cell will have a similar type of a structure, right? So it'd have its connection to the cytoskeleton, and they would have a receptor that kind of reaches out and connects. And so what these two different cells do is basically they're gonna have these extracellular connectors. So what they do is they basically kind of grab these two receptors, kind of grab on, they kind of like grab hands. It's like a train hitch, like coupling trains, right? So they basically have both hitches and they kind of hitch onto each other. In this case, calcium is part of the story. But the important thing to know is you got a good connection, but you also have some space here for some communication. Right? So this is kind of like the intermediary between the two. As a matter of fact, I always like to kind of think of this by analogy as basically buttons, right? So buttoned up, like a buttoned up shirt. If you connect two pieces of fabric like a shirt with buttons or a coat, right, there is some open spots in between the connections, right? So you have nice connection, first of all, but you have a little bit of space in there. So like if I had an internal pocket or something like that, then we'd reach in and grab. If I've got buttons, I could slip my hand in between the space there and grab whatever I have in my internal pocket. So notice I've got a compromise between connection and free space, communication, right? So the extremes are represented by the tight junctions, which maximize connection and have minimal communication. And basically what a tight junction is basically like two proteins that are literally just fastened together. So these guys are tight. I mean, there's no space in there. So there's no communication at all. This is really good if what you want is no leakage between these sheets. Think about it. Let's take a look at our clothing again, right? Let's imagine that I've got a coat on and I've got it buttoned up. It's really windy. It's like -10, and like the wind chill is like really polar. So the air is really cold and it's windy. So it's like getting down to like wind chill. It's like getting down to -30. Do I want to have my coat buttoned up? No, because the wind can get into the space, right? And I can get cold. So what do I do instead? I zip it up. So this is the zipper. So when I zip it, then there's no space in between those two pieces of fabric. That creates a no leakage or an impermeable barrier so that the wind does not get into me. Okay. So that's perfect. But guess what? I let go of. I can't reach into my inner pocket now, can I? So I've had to give that up. To stay warm, I've had to give up on reaching for my cell phone, right? So, but that's necessary if it's -30, because you probably shouldn't be dorking around on your cell phone when it's -30 anyway. Just get from point A to point B, right? One warm building to another warm building. You can do your cell phone stuff later, right? So that's one extreme. The other extreme is communication. So here, basically, what's going to happen is you're going to maximize your communication and you're going to minimize your connection. So you have two different types of communicating junctions. You have gap junctions in animals and plasmodesmata in plants. And so here's what it looks like in animals. So basically, when you have a gap junction, what you have are two cells that are connected by these like little channels, these gaps. So that what happens in cell number one in the cytoplasm will immediately rush through to cell number two. So what happens to you happens to me simultaneously. Now, the reason why you would do this is to get coordinated response. So if you need a group of cells to act in unison or in coordination, you connect them with gap junctions to facilitate communication. The perfect example of this is your cardiac muscle cells, right? Because unlike skeletal muscle, which doesn't contract all at the same time, skeletal muscles actually rotate when they contract. And they can also contract partially. Typically, your skeletal muscle only contracts enough to clear the load, right? So you don't contract continuously. Oftentimes, if the load is fairly light, then your muscles there'll be a lot of uncontracted musculature in your skeletal muscle because you have more capacity to lift the load, right? And so often your skeletal muscle is very, I don't say inefficiently, but incompletely contracting. That's what it's designed to do. Your heart muscle is different. Your heart muscle has to contract in totality, completely, and it has to contract in unison, right? So it can't kind of do this ripple effect like muscles do. It can't rotate like muscles do, because then what would happen is you would get rotation of contraction through your heart, and it would look like a quivering heart, which is death, basically. You get out to paddle time and zap you, right? That's what that does. So what they have to do is all the muscle cells have to basically contract all at once to give you a really strong, powerful pump so it can shoot the blood all the way to the periphery of your body. The only way you can do that is if all of your cardiac muscle cells get the same message at the same time. That is, they all hear the contract message at the same time. So like if I wanted you guys to all do the same thing at the same time, like say, for instance, I wanted you to jump. I say, okay, on the count of three, everybody jump. 1, 2, 3, right? You could do that. Why? Because you're all interconnected to the same airspace. You all heard the instructions at the same time as it was given. Gap junctions are essentially the equivalent of all the cells hearing the message at the same time. The only way you can get that coordinated, unified contraction is if all of your cardiac muscle cells are interconnected with each other through gap junctions. Maximum communication. So here's kind of the pictures of what they look like. You can see you have the tight junctions, which are very, very tight. So they're fastening everything together. So you can see how basically they kind of pull together and they're kind of battened together. Very, very tight. And then on the other side, you can see the communications junctions where you have your gap junctions. This is really cool, right? Because when you talk about a gap junction, you're not just talking about an occasional hole here and there. No. You're talking about a ton. Take a look at how many are there. Can you count how many little holes are there? No, it's incredibly porous, right? I mean, it's like a ton of little holes in there that are interconnecting these two. I think this is a cross-section of intercalated disc, which is the boundary between two cardiac muscle cells. So you can see that you're not just talking about free-flowing cytoplasm like through just a couple of holes. You're talking about a ton of holes that accommodates a very like immediate, like literally being in cell one is almost identical to being in cell two. It's like being in two places at the same time because it's just so open between the two. And then of course, those are two extremes. And then you have the adhesive junctions, which have an infinite number of variations in between those two, right? So you could have adhesive junctions that are a little more connection oriented. You can have adhesive junctions that are a little more open and a little more communication oriented. And you can have everything in between. This is the reason why adhesive junctions are the biggest junction and the most common junction we have in the body. For instance, this is an example of a desmosome, where you can see one little protein pad on the inside of one cell, another little protein pad on the inside of the other cell. You have some cytoplasmic connectors to connect to your cytoskeleton, and you have some intercellular connectors to kind of grab onto each other's pad. Okay, notice you also have intercellular space in between. That gives you a little bit of free flow. Can you loosen that up to give you some more space so you can be more communicative? Sure you can. You can choose a different type of an adhesive protein, right? And still maintain your connection, but just kind of loosen up a little bit so that you can talk a little more freely to your neighbors. Okay. So this is the most common type, this adhesive junction strategy. Let's take a look at the plant cells, right? Give the plants a little bit of love. You can see how this goes, right? So we talk mostly about animals, and then we kind of just give the plants a little lip service. So it's like, oh yeah, and then there's plants too. Right, but this is actually kind of important, right? Because plasmodesmata are actually these like little canal bridges that are actually crossing over the cell wall, connecting one plant cell to another plant cell. In this case, instead of connecting your cytoplasm, you actually take the smooth ER of one cell and you kind of ooze it, or you kind of squirt it through that little plasmodesmata so it pops out on the other cell. So in a sense, two cells are actually sharing a little bit of their smooth ER. Because everything else is different. And the rest of the smooth ER is different as well. Right? So all they're really sharing is this one little piece of the smooth ER. And the idea is, like anything you wanted to communicate there, you could like fuse different vesicles to this thing, and all those little proteins would come on over here, and then you'd be able to move back and forth freely between these, just using vesicular traffic. So if you think about it though, all the plants, and this isn't just like all those plant cells you guys saw with the Elodea or with the onion cell, those are all interconnected, which makes a lot of sense though in retrospect, doesn't it? Think about it. Here's a plant, I just gave it some water. The roots of that plant are made out of cell walls, right? So how is water supposed to cross these really tough structural cellulose cell walls and get into the roots, and then get from one cell to the next, to the next, to the next as it goes up to the plant? You see the problem? The cell wall, which creates a very, very strong protective barrier, can also kind of cut against you as well. So how do you then move stuff around the plant in an efficient manner? How can you move like water and different solutes and sugars and things like that throughout the plant in the places of need in a very quick and efficient way? Well, the plant strategy, interconnect all of our cells with plasmodesmata. So this basically facilitates the transfer of fluids and minerals and sugars and things like that from one part of the plant to the next part of the plant, and eliminates the barrier component associated with the cell wall itself. Now, this becomes important, like if you take a plant physiology class, you'll get much in, because that whole fluid, how plants circulate fluid, and that's a big thing in plant physiology, and a lot of it is created by the plasmodesmata, by the fact that all the cells are linked together. I was like, yes, finally, the end of 4, and that is the end of the chapter for coverage purposes. So that'll be the exam right there.
Is it going to be the rest of 3 and then 4?
Yes, the rest of 3 and 4. So we'll give a healthy start on 5.
This is like shirtless running down 16th Street. You screaming, I'm
You got get down Spear, right? So we can like.
7th on St. Patrick's Day. That night.
That's okay. We won't judge. We'll just let it all go. Okay.
Professor Puss.
Yeah.
On that chapter. Going into 13th then on that?
For what?
There we go. Because 12 is just one.
We're not doing genetics. I believe it's just, does anybody have the schedule? Well, I've got it up here.
It says 11 and 12.
Does it say 11 and 12? Well, so mitosis is actually pretty short. That might be the reason why they're bundling it up.
It says it's for 80 minutes. I'm just kind of straight.
So let me double check just so we can clarify that. So there's a question on the table for lab. Which labs are we covering? Basically, is the question. Yes.
Yeah.
And I'm just going to pull up the lab schedule because the department sets the schedule, not me. Yeah, those are both easy. So mitosis and chromosome squash, those are both easy. So 11 and 12.
Okay.
They're pretty straightforward. Yeah.
I just didn't know.
Yeah, it's not genetics.
Yeah.
Genetics takes a lot longer to get in there and kind of, so it's a little bit more than that. So. Okay. No rest for the weary, chapter 5. That's just the point of the semester we're in, right? This is the gut it out and grind it point of the semester. So chapter 5 is all about membranes, right? So we just talked a little bit about all the organelles that are in their functions and what's going on with the organelles. Now we're going to be talking about the membranes themselves and focusing there. Next week is spring break, correct? This is week 9. Next week is spring break. So don't show up because I'm not going to be here. I may be actually right behind Chris running down Spear Boulevard. That's okay. That's just occupational hazard. I'm used to that. Yeah, I'm used to that. So let's take a look at and refresh our memory. There's going to be some review stuff in here, right? So from our chapter 3 stuff of it, right? So we want to take a look at our membranes and the structure of it. So we already know that we have a membrane composed of phospholipids, and then it forms a bilayer. We also already know that there are proteins embedded, so they're inserted into the lipid bilayer. We saw one already, right? We saw integrin embedded in the membrane hanging onto its extracellular matrix, right? We saw that. So this basically makes, when you insert a protein into the lipid bilayer, it's a type of protein that we refer to as an integral protein, integrated into the membrane itself. So all of about, oh gosh, 50s, 60s or something like that, we came up with a model to explain what's happening at the plasma membrane. And we call it the fluid mosaic model, okay? And the fluid mosaic model has 2 pieces to it, the fluid piece and the mosaic piece of it. But what we actually, the way we envisioned this was with a phrase, and this was actually a phrase used by one of the cell biologists at Anschutz, actually. And it's a pretty common way that we thought about it. She was like a big kind of cell biologist there. We refer to as rafts on an ocean. And what I want you to do is I want you to kind of consider that phrase, right? Think about that phrase a little bit and let that conjure an image in your head. Rafts on an ocean. And I want you to tell me what exactly is it that you're seeing? What does your image look like when I say rafts on an ocean? How big? A dinghy? A dinghy would be actually a pretty an upgrade from a raft, right? Because it actually has a hull, right? I mean, what is a raft? Not a white water raft, right? That's our sickness because we live in Colorado. We're talking in the ocean.
I think it's one of those things you sit in. Or is it a canoe?
A canoe,
Doesn't it look are we talking like you're stranded?
like a raft. Think Castaway.
It's like
Yeah, where do you see a raft?
like an octagon shape.
How many of you guys have seen the movie Castaway? What did that look like? Well, he was floating around on what was it made out of?
Is it the one where your guns? No.
What wood, right? Yeah, where did he get the wood from? Yeah.
Thinking of the. Didn't we have, wasn't there a scene before that?
Wilson. It's the one with Wilson.
Isn't there the scene
Yeah.
in the first Crashes he gets the
Yeah,
boat?
Castaway. Yeah.
He makes the raft too.
So what did he make it out of? How big was it?
Didn't he make it out of a porta potty?
Yeah, it was basically a makeshift situation, wasn't it? Is this something that you would want to be on in the middle of an ocean? But if you had spent, 4 years with Wilson as your best friend, maybe you're going to give it a shot, right? So how big is a raft? Yeah, actually, I mean, if you've ever done a look at the history of rescue, like sea rescues and things like that, I mean, pretty much a raft is whatever floats and you can hang on to. One of my other sections mentioned the door from Titanic, right? That what's her face was on while the dude froze to death. Yeah, Rose. Yeah. When she was old, she died in the movie, but she was old, right? Yeah, they could have both. I like that. They both could have fit. She's selfish. Yeah, that's brutal. But so basically, it's whatever you can make it, right? So how many people can fit on a raft? 20? 1, maybe 2, right? Okay, now we got that. It's basically a bunch of scrap wood that you lash together with vines, and it's small. Got it. Okay, now let's take a look at the ocean. What does this look like in the context of the ocean? Imagine you are in the rescue plane. You're flying over the area of the sinking, and you're looking for survivors. Yeah, what are you mostly seeing when you're in that plane? Like an endless horizon of water, right? Which is the reason why a lot of times the rafting thing doesn't work, because you're so tiny and nobody's ever going to see you, right? Yeah, you have to have something to sort of make yourself stand out. Think about that. That's exactly the way we thought about the membrane. We knew that there were proteins out there. We just talked about the integrin, right? But here's a couple of things. Number one, the way we thought about it was just like a raft on an ocean. We thought that there was very few things out there, and what they were was small, and they were floating around in an expanse of membrane like the ocean. Vast expanses of membrane in between these little itty bitty isolated protein rafts floating around in this ocean of membrane. And the other thing we also thought as well was a raft. Do you get on a raft and you say, look, I'm going to go due east? No. What's happening to you on a raft? You're just going wherever the ocean takes you, right? So there's a sense of randomness to it, and you're just hoping you get lucky. Knowing me, I'd get on a raft, I'd go through all the pain and heartache of beating off the sharks and all that sort of stuff, and I would probably end up drifting to another deserted island, right? With fewer coconuts, or something like that. So that would be me. That would be my luck. So the idea is there's a sense of randomness. So these proteins are actually floating around, and they're randomly floating around in this ocean of membrane. That's kind of the way we thought about it. As it turns out, it wasn't quite that way. right? That was our original thought. But we had to modify that. So we have an ocean made of phospholipids, yes, and this is our phospholipid, so I'm not going to belabor this one. So you already know that you have a phosphate head and you have these nonpolar tails, and that's basically what gives us our structure, our lipid bilayer, right? So when we start to connect this together then to our proteins, we see we've got a number of proteins in here. So you can see you got a protein over here that's inserted into the membrane. That looks like it's some sort of a channel, or it looks like something's designed to go through that little pore. So that's one type of protein. You can see other types of proteins doing several different types of things here. Some of these are inserted in the membrane. Some of these guys are not, like this guy right here. That would be like a peripheral protein. He's not in the membrane, but he's associated with a membrane protein. Same thing with this guy here. He's not in the membrane, but he's associated with the membrane protein. And then this is the membrane protein right there. There's a couple of guys we notice here. This guy right there, that's an integrin, right? We just saw that one. Here's your fibronectin. Attached in this case to your extracellular matrix. And then these guys here are your glycoproteins. You can see these little carbohydrate branches that are part of your self-sorting and cell identification. And then of course, you have your glycolipids, which is also part of your cell-sorting and cell identification. And then one of the things that's kind of new in this image is this guy right here. What is that? That's cholesterol, right? You can see that four-fused ring, right? That classic steroid, which is a lipid, four-fused ring. We'll take a look at the function of cholesterol in your cell membrane in a little bit. But you can see how everything is tethered to or associated with your cytoskeleton. So you can see your cortical-actin underneath here. And then your intermediate filaments down here kind of creating the scaffold. So you can see this guy is bound to an intermediate filament and tethered to the membrane protein. So everything is well anchored and well connected. So let's take a look at some of the components of the cell membrane. So some things we know well, right? First of all, the phospholipid bilayer. We've seen that one. We're pretty familiar with that one. We're pretty comfortable with that one. It's very flexible because it's just a phospholipid bilayer, but it's a great barrier to permeability. That's a good thing. If you want to keep stuff out, that's good. But in it is also transmembrane proteins, right? These are what's called integral proteins. Basically, they're inserted into the membrane itself. Now, there's a little bit of nomenclature that I want to clean up that oftentimes throws students for a little bit of a loop here in this particular case. And it's the idea of an integral protein. So an integral protein is basically any protein that is inserted into the membrane, right? And the way they do that is with transmembrane domains. Integrin is a type of integral protein. So a lot of times this confuses students because they'll see integrin, integral, the two terms are very close together. It'll kind of confuse them a little bit. Always remember, integrin is an integral protein, but not all integral proteins are integrin. That make sense? Let me say that again.
Yes.
Integrin is an integral protein, but not all integral proteins are integrin. So integral proteins represent all proteins that are inserted in the membrane. One of that family of proteins is called integrin. But there's other proteins that are not integrin that are still integral, like the channel, for instance. So for instance, all of these are integral proteins, but only one of them, this guy over here, is integrin. That makes sense? Now, because of that confusion, I generally don't use the term integral. I usually use what we know, which is transmembrane, because transmembrane domains are required in order to integrate or insert into the membrane. So instead of saying integral proteins, I usually say transmembrane proteins. So in addition to this, what you're also going to see out there is a broader network. So these are your peripheral and associated proteins that are not integrated, but they're groupies, right? They're kind of hanging out with the cool kids, and they're just kind of like basking in the afterglow of coolness, right? If I can't be as cool as the transmembrane proteins, then I'll hang out with them, and that'll kind of give me some cool cred, right? Doesn't really work, actually. Just saying that doesn't. Most people know the groupies, and it comes across more pathetic, really, than impressive. But anyway, that's that. And then, of course, you have cell surface markers, right? So this is your profile, your glycoproteins and your glycolipids. So that's what's out there. So this is like, I'm a liver cell. I've got all of the glycoproteins and glycolipids that sort of give me that liver cell profile, right? So that's how everybody knows who I am, okay? Along with the glycoproteins that are your self-tags. Was that a question? Yep, there's tons of glycolipids out there. Every single cell has a spectrum of glycolipids. Yes, everybody has the same MHC, but different cells will have a different type of glycolipid and glycoprotein collection that's specific to their cell type. Exactly. So a neuron would have completely different glycoproteins and glycolipids than a liver cell. And so this is for you table people, right? This is your components basically just put into a table. That's all it is. So that's for those of you like tables. Okay, so let's take a look now at the nitty gritty of this fluid mosaic. Because you talked to me about this whole fluid thing, that's fine. That basically is the fluidity of the ocean, right? Water is fluid. The raft is floating around on a fluid ocean. And then you're talking about this mosaicism, that there's a lot of proteins out there. And that's nice, right? But it's all blah, blah, blah. I hear a bunch of words. I'm not convinced. I can make stories up too, right? If all you do is make up stories and you don't prove them, it's called philosophy. Ooh, that hurt. Sorry, Kurt Pond, if you're listening to this, no harm meant, right? I love Kurt. You guys had him for philosophy. He's going to retire, so if you haven't had him, I would suggest you take him because he's great.
Older guy?
Likes to wear hats a lot. Yeah, he's really cool. He's a really great guy. Good soul. Gonna miss him when he retires. We're like, no, not Kurt. But I guess that's what's happening. So that generation of professors that brought me in, they're now starting to retire. So what does that say about me? It's like you guys are all like raising your eyebrows. You're like, yeah, you're the only one in the room who's not aware of it. But anyway, no, I am aware of it. Trust me, I have to live in this body. But anyway, so prove it to me, right? Okay, let's prove the parts of the fluid mosaic model. Let's start with the mosaicism first. So this is the experimental proof. Of mosaicism. It's called the freeze fracture experiment. And here's how it works. Typically, you take a cell and you embed it in some freezing compound, and you basically jam a knife in there. It's pretty brutal. It's kind of thuggish a little bit. And all you do is give that knife a twist and you sort of crack it open, essentially. Now, eventually, you're going to get a pretty favorable crack or fracture pattern. And I had one of my graduate student friends actually was doing some of this work. They're trying to get a cross section, a cell cross section, so they could study it. And they had to do this. And I mean, you have to do like potentially hundreds of fractures to get a good slice that shows you what you want to see. It can be quite a bit. So there's a lot of patience and persistence associated with this particular technique. It's somewhat brutish, but when you get it, it's really nice. And so in this case, we get a nice favorable crack. And basically what it's going to peel off the top portion of the bilayer, leaving the bottom portion underneath. And then if you zoom that up, you basically have the top portion with all these proteins in it. And here you can see like a little channel that was in there. It kind of came out with the top portion. And you can see like the underneath portion. So you can see two things. First of all, you can see proteins that go all the way through. You can see it from the top. And you can also see proteins that only go part way through. And you can see them sticking out of like little nubbies right there, right? So they kind of like are kind of like that. So you can see both. And then basically what you want to do is you want to take a look at this with electron microscopy and then see what you see. What we thought, remember the rafts on the ocean, we thought mostly ocean, open water, an occasional dinghy here and there, right? What we saw instead was this. A ton of stuff out there. It was mind-numbing. Now, the projection is not as good. It's a little bit better here than it is in 1019. But what I'm going to do is I'm going to put like a little red spot. So I can see a little bit more detail, a little more resolution here on this image. And I'm going to put a red spot on the peak of each one of these dimples. Each one of these dimples represents a protein that is inserted into the membrane. And I'm going to basically put a little red dot on all of the big ones and little ones that I can actually see, just to give you a sense of scale. Okay, here we go. Okay. Are you getting the idea? Do I have to keep going? You're like, yes, you do. You just did this to us in chapter 4, and this is our payback. We're making you do the whole thing. It's a whole cluster of them down here. Okay, so you get the idea? So that just keeps going and going and going. So that's big dimples and little dimples and little dimples in between, and ridges where there's like 3 or 4 dimples in a row. And so you can see that what you're talking about is not just rafts in an ocean, are you? So you're gonna have to modify it. So the scale is like astronomical. Kind of blew our mind a little bit. We're like, whoa, wait a minute. We were like way off, right? So we're not talking about rafts in an ocean, the occasional easily missed raft floating around in what's mostly open ocean. What we're talking about is a busy marina in Miami during the apex of boating season. Does anybody know what the Miami Marina looks like? Can you see a lot of water in that marina? No, right? Just watch an old episode of Miami Vice. Tell me how much, you see the thoroughfares, right? So the boats can get in and out of the marina. You can see that, that's water. But when where the boats are lashed up, I mean, you basically see the walkway, and it's just basically, right? Starboard, stern, starboard, stern, starboard, port, starboard, port. I mean, it's just side to side to side to side to side to side. I mean, you pretty much don't see any open water. All you see is just boat. You can literally just walk from one deck to boat to the next deck and never actually see water. You know it's down there because you can feel yourself floating. You know it's down there somewhere, but you just can't see it because it's just literally like just packed with boats. That's basically what we saw here. So the mosaicism isn't just an occasional thing. We're talking about just basically a massive packing of proteins that are out there. So that kind of basically proved to us, oh yes, it is a mosaic. And not only that, but the scale of it is astronomical. So we're talking about a ton, not just a few proteins out there. We're talking about a ton of proteins out there. So that brings up a big question, right? So if there's so many proteins out there, then what are they all doing out there? Think about it. If you're out there in numbers and you've got jobs to do, then what does that do about your view of the balance of work being done by the cell? Yeah, right. So it kind of reorganizes the way you think about how work is distributed in the cell. Okay, now let's work on the fluid piece, right? So we covered the mosaic piece. Now let's work on the fluid part of it. So just to refresh our memory, right? Phospholipids basically are resting on a 3-carbon backbone, the glycerol backbone, with 2 fatty acids that are hydrophobic and a phosphate head that is hydrophilic. And we know that we get our phospholipid bilayer for free, right? But remember, this center portion here is nonpolar. What does that mean? That means that if you're polar like water, guess what? No dice, you're out, right? If you're polar like sugar or salt, no dice, you're gone, right? If you're charged like sodium or potassium, sorry, no dice. But wait, what if you're a lipid? Like a steroid, for instance, like estrogen. Well, you can go through, which is the reason why the estrogen receptors are not on the outside of the cell, they're inside the cell. Because those lipids, those small lipids, and small molecules like oxygen and CO2 can skitter through because they're non-polar, so they can go through. But here's the problem. Are you going to be able to survive long if you can't get water inside you? How about salt or sugar? Yeah, you got a problem there, don't you? So you got to fix this. So this is what we need. It's a good barrier, but it's too good. So you got to fix that. So this basically is the evidence for fluidity. We'll talk a little bit about fluidity. So this is our fluidity experiment. It's called the cell fusion experiment. So what we're going to do is we're going to take a human cell with membrane proteins that we can label blue in this case, and we're going to fuse it to a mouse cell with membrane proteins that we can fuse, we can color as red. Okay, so we can distinguish between the two of them. Now we have a testable prediction. If it's fluid, notice the if then. If it's fluid, then I would expect it to intermix. That is to say, I would expect something to look like this. So all the human cell proteins that are over here and all the mouse proteins over here should start to move back and forth and scatter, basically, randomize. If it's not fluid, then I shouldn't see any intermixing, which basically means when I fuse this together, the human proteins will be over here on the human side, mouse proteins will be over here on the mouse side, and then I'll have like this line of demarcation in between. So what did I do? I labeled my proteins, I fused the cells, and then what did I see? What I saw was mixing. So the human cells were getting all the way over to the mouse side. The mouse cells were getting all the way over to the human side. How did they do that? They had to move. That is to say, they had to drift. Just like if a raft goes from here to over here, how did it get over here? Because the water underneath it had to be fluid enough to move it around. That basically means the fluidity means that the water molecules are shifting around each other. They're sort of mixing around each other. And as they mix, that's what creates drag on the underside of the raft, which is the reason why the raft drifts. Drifting is the consequence of that mixing effect of water molecules dragging and scraping along the bottom of the raft, causing the raft in very small degrees to gently move as the water underneath it is mixing around and pulling it in a particular direction. So cell fusion experiment basically proved to us that not only is it mosaic, but it now is also fluid as well. So we do have proteins floating around in the actual membrane itself. So there's one little piece of fluidity that I want to cover, but we're at our witching hour, and I want to take a little bit of time to unpack this one because it's going to apply what we learned before in chapter 3 with our 4 rather, 3 with our margarine effect. Remember the margarine effect? So we're going to apply those principles of lipids to our membrane. We'll do that next time. Yeah.